Well, Sclerotinia stem rot is probably one of the key disease issues that producers in Western Canada face. So it is an issue that not only affects canola, but also most of the broadleaf crops that we grow, and as well as the broadleaf weeds. So it is a disease that's driven by moisture and temperature, and it affects canola primarily at the flowering, at the time of flowering, and will use the petals as a carbon and nitrogen source. So the fungus produces spores from the mushroom-like structures or apothecia that it produces. Those are released into the air. They'll land on canola petals while they're still up in the flower. And then when the flowers or the petals lodge, they'll lodge in leaf axils and bases. And the fungus uses the petals as a carbon and nitrogen source. It's also a really nice environment because often you'll see some rather nice large water droplets that form in that area. So it's a good environment for the fungus in terms of infecting the plant. That's a good question. The last few years, certainly, we've seen some pretty significant levels of stem rot in fields across the prairie region. Maybe not so much in some of the drier areas of Manitoba this year, but it's a disease that, really it comes and goes. It really follows the weather. So depending on what the weather is like, starting in about mid-June through the end of June and into most of July, that will really drive the disease. So if it's a drier than normal year, in many cases the disease will be very limited. So the objectives were really ultimately to look at trying to come up with some better tools for producers and consultants or agronomists that are advising farmers to decide whether to spray or not to spray. So to figure out what the disease risk is, and it actually is based on some work that I did as a grad student in the 80s in Saskatchewan, where we looked at using the percentage of petal infestation, so the percentage of petals that were infested with the Sclerotinia fungus, and whether or not that level of infestation was very low, medium, or high, and that would indicate the potential risk that the crop was at. The issue with the current way of doing that, it's an agar plate test, which is still functional, but it usually takes anywhere from 5 to 7 days for the test to be done and the results to be read. And it's something that probably for a lab professional, a seed testing lab, or a pathologist like myself and the technical staff that work with me, could be done fairly easily. For a producer, or especially an agronomist that's advising a producer, if they've got 10 to 15 clients who all have fields of canola, I would say it's almost impossible for them to do all the testing and picking of petals and plating and so on. So the project was looking at a better way of assessing the level of petal infestation, and that's using quantitative PCR. Well, it differs from the current agar plate test in that you're using more modern DNA detection technologies, specifically quantitative PCR. So that's a test that once the samples are received and processed, the results can be available within a matter of hours. So it's something that there's a fairly quick turnaround time on. With petal testing, whether it's with qPCR or with agar plate testing, it's a way of assessing whether the pathogen is there and at what level. But with Sclerotinia, it's a disease that you really need to look at all three components of the disease triangle. So is the pathogen there and at what level? Is the weather favorable? And next, is the host at the right growth stage, so i.e. flowering? Do you have a reasonable yield potential? Because if your yield potential in your canola is much less than about 35 bushels per acre, probably spraying for Sclerotinia is not going to give you a response that is going to be economical. Well, what we've done, we brought on staff a very nice young lady, Barb Zeisman, who's the master's student at the U of A that started probably about a year into the project. So what we did in the meantime was colleagues in Saskatchewan, Alberta, and Manitoba that were collaborating on the project, collected petal samples, in some cases did some agar plate testing, and then went back to the same fields and assessed the incidence and severity of Sclerotinia. We then, when we took petal samples, we also collected flowers that were then frozen and kept for Barb to process once she developed the qPCR technique. So the first part of our program was really getting the qPCR technique up and running, and they've done that. It's specific for Sclerotinia sclerotiorum. The results indicate that there's no cross-reaction with other species of Sclerotinia and with other fungi that you'd normally find, let's say, floating around in the air in July in Western Canada. So the test is specific. It's also quantitative. So she was able to develop a test where she could look at the level of DNA of Sclerotinia. So right now, she's working through all of the flower samples that were collected and frozen the previous 3 years. And what's happened is that we've actually got an extension on the project through the Ag Funding Consortium in Alberta, and I think the Alberta Canola Producers Commission and other canola producer commissions, to extend the work another 2 years, so that to look at further refining the qPCR. So there's some internal controls that she's put in place, and doing some further field testing of it. And then ultimately working with some of the private sector seed testing labs that have that capability, because ideally it's a test that could be shunted into the private sector. It's work that could be done at a time of the year when I think their workload is pretty minimal. So it might be something that would give them some value as far as giving them something to do and utilizing the equipment and the expertise that they have at a time of the year when things are fairly quiet. Certainly in terms of assessing the level of petal infestation and doing it quantitatively, it is an improvement over the agar plate test, because the agar plate test, there is some art and science to it as far as identifying Sclerotinia on the culture plates, so there is some potential there for misidentification. The other thing again is the time frame that you're looking at with agar plate testing, whereas with the qPCR, it's very accurate as far as identifying Sclerotinia. It's quantitative, and it's something where the results can be returned back to the producer or the consultant within a matter of a day or so. So it would give them a very rapid turnaround. They would know what inoculum levels are there in the field. At that point, then they'd need to look at what is the crop yield potential, what's the growth stage, and in terms of what are the weather conditions like. And with that set of information, those 3 areas, they can make I think a fairly effective call as far as what they would need to do in terms of a fungicide application. Well, certainly in terms of allowing producers to get a better handle on what inoculum is floating around in the air, in terms of risks from Sclerotinia. To be honest with you, from a research point of view, it gives us a tool that can be used to better understand the epidemiology of the Sclerotinia fungus during the growing season. So as a pathologist, we can go out and do some more intensive sampling, finding out exactly when the fungus is out in the air, the levels, how those levels change with flowering period and weather conditions and so on. So that's the idea. So if you look at current technologies, or let's say current diseases, so Fusarium graminearum, for instance, you have a number of labs that do PCR testing of grain samples or other plant tissue samples. If you look at clubroot and canola, there are labs that will do commercial testing, and I think in some cases qPCR testing or just standard PCR testing of soil samples. So this is something that a lot of the labs either have the current capability of doing this, or they're certainly looking at, they're doing standard PCR, but they're seeing that there's value in expanding their ability to look at quantitative PCR. So it's something that would lend itself to being moved into the private sector.